What if there was a compound that could help you live longer, healthier, and more resilient to the challenges of aging? Meet Astragaloside IV, a fascinating molecule derived from a traditional Chinese medicine herb called Astragalus membranaceus. Known for its potential in health and longevity, Astragaloside IV has been gaining a lot of attention in scientific research due to its various beneficial properties. As you can see on this figure, several studies have shown that Astragaloside IV has demonstrated antifibrotic, neuroprotective, antioxidative, antitumor, and anti-inflammatory effects, both in vitro and in animal models. In this video, I will explain what makes this compound so special, the science behind its benefits, and whether it could be a supplement for better health. But let's start with the molecule itself. Astragaloside IV can be found in Astragalus radix, which is the dried root of Astragalus membranaceus. This plant has been used in traditional Chinese medicine for over 2000 years. There are various astragaloside family members, which chemically are all triterpenoids and are responsible for the medicinal properties of Astragalus radix. From the four astragaloside members, Astragaloside IV is considered the most active member. As already mentioned, Astragaloside IV demonstrates a wide range of pharmacological activities, which I will discuss on the next slides. Please note that I will only show a few selected papers with some data snippets. This is by no means an exhaustive literature and data review, so I would highly encourage everyone to check out the papers I have listed. You all know that longevity is a pretty hot field these days, and Astragaloside IV was studied for potential longevity effects through telomerase activation. Now, what are telomeres? Telomeres are protective caps found at the end of our chromosomes. They are consisting of repeated DNA sequences, and they act like buffers and shield our genetic material from damage during cell division. However, these caps can't be fully replicated each time a cell divides, and as a result, telomeres gradually shorten, which contributes to cellular aging, reduced tissue regeneration, and the higher risk of age-related diseases. Fortunately, our cells have a natural defense, an enzyme called telomerase. Telomerase can extend telomeres, helping maintain chromosomal stability and supporting cellular health. Now, in a study from 2014, it was shown that cycloastragenol, so astragaloside without the sugars attached, stimulated telomerase activity dose-dependently, both in human keratinocytes and in rat neuronal cells. The authors, and this is not shown here, also did an in vivo study where they administered 100 mg per kg of cycloastragenol for 7 days before doing a few behavioral studies, from which they concluded that the compound also attenuated depression-like behavior. In another study, they treated nucleus pulposus cells, so the cells of the jelly-like substance in the middle of the intervertebral disc, with high glucose to induce stress, senescence, and apoptosis. Treatment with both cycloastragenol and Astragaloside IV increased telomere length and ameliorated the telomere attrition. Astragaloside IV has been studied for quite some time for its anti-inflammatory properties, and here I'm just highlighting a few of the many papers that have been published on this. In a 2023 paper, Astragaloside IV was studied for its potential therapeutic effects on COPD, so chronic obstructive pulmonary disease, and the authors showed that in vitro, Astragaloside IV suppressed the expression of CXCR4, ROR-gamma-T, IL-17A, as well as Th17 cells, and at the same time promoted expression of FOXP3 and IL-10, as well as Treg cells. So here you just see a figure from the paper showing that with increased concentration of Astragaloside IV, the IL-17A mRNA levels decrease. In vivo, Astragaloside IV alleviated Th17 Treg imbalance induced by cigarette smoke. So in this mouse model, you see that with cigarette smoke, the number of Th17 cells go up, whereas with dexamethasone, which is a corticosteroid that is also used in the treatment of COPD, they can be pushed down again, and also with Astragaloside IV here. In another study, it was shown that Astragaloside IV regulated differentiation and induced apoptosis of activated CD4 plus cells, so T helper cells, in the pathogenesis of experimental encephalomyelitis. Finally, in a study published in Phytomedicine, it was shown that in a DSS-induced colitis model, Astragaloside IV treatment effectively prevented and alleviated symptoms and at the same time reduced levels of pro-inflammatory cytokines such as TNF-alpha, IL-1 beta, and IL-6. So here's the high-dose group, and this was 100 mg per kg, and you see that clearly the score is reduced. Excess accumulation of reactive oxygen species or reduced antioxidant capacity leads to an imbalance in the oxidative and antioxidant systems of the body. These changes result in oxidative stress, which in turn causes inflammatory neutrophil infiltration, increased protease secretion, and oxidative intermediate generation. Astragaloside IV is known and studied for its antioxidative properties, and I wanted to highlight two studies. In the first study I wanted to highlight that was published in Cell Cycle in 2022, it was shown that Astragaloside IV significantly improved cardiac function and reduced harmful changes after myocardial infarction, such as fibrosis, so scarring, and hypertrophy, so thickening of the heart muscle. Here you can see the effect of Astragaloside IV in mice on the fibrotic area. And here you can also see an additional effect that you actually reduce cleaved caspase. So this indirectly shows that Astragaloside IV can suppress the NLRP3 caspase-1 gasdermin signaling pathway, which is linked to inflammation and cell death. In another study, Astragaloside IV significantly improved endothelial dysfunction in diabetic rats by enhancing vascular relaxation, increasing nitric oxide production, and boosting eNOS expression. It also reduced oxidative stress by lowering ROS levels and restored antioxidant enzyme activity while inhibiting calpain-1 activity. These findings suggest that Astragaloside IV protects blood vessels in diabetes through antioxidant and calpain-1 related mechanism. So let's have a look at some of the figures from the paper. Calpain-1 is a calcium-activated protease, and when overactive, it drives pathology, and especially the high dose could actually lower significantly the calpain-1 levels in this experiment. Then you can see the reduction of the ROS levels. And finally, eNOS stands for endothelial nitric oxidase synthase, and it synthesizes nitric oxide from arginine, which helps with vasodilatation. It inhibits platelet aggregation and also adhesion, and reduces leukocyte adhesion to the endothelium. So enhancing eNOS levels, which is shown here, is actually a good thing. A systematic review and meta-analysis of 23 animal studies found that Astragaloside IV shows robust antifibrotic effects across lung, heart, liver, and kidney models. Now let's have a look at some of these. In this first study from 2019, published in Scientific Reports, the authors showed that Astragaloside IV inhibited hyperglycemia-induced endothelial mesenchymal transition of podocytes, so the cells of the kidney. Now, TGF-beta is widely recognized as a major inducer of EMT. In this figure, you can see that high glucose levels leads to increased TGF-beta levels, and these levels can be brought down with increasing concentrations of Astragaloside IV. In the study, they also showed that Astragaloside IV enhanced autophagy and increased sirtuin-1 expression. Sirtuin-1 is a histone deacetylase, and generally, increased expression is good for preventing age-related diseases and metabolic diseases. Now again, in this figure here, you can see that high glucose leads to decreased mRNA levels of sirtuin-1, and they can be brought back with high concentrations of Astragaloside IV. In another study, Astragaloside IV significantly improved survival for Coxsackie-induced myocardial damage and fibrosis. So here you see percent survival of the mice with the virus. It actually goes down, and it's brought up again with Astragaloside IV, so increasing survival rate. Specifically, Astragaloside IV protected the mice against myocarditis characterized by increased body weight, decreased serum level of creatine kinase and lactate dehydrogenase. It also suppressed expression of interferon gamma and IL-6 in the heart, and enhanced systolic and diastolic function in the left ventricle. At the pathological level, Astragaloside IV ameliorated myocardial fibrosis, as shown in this figure here. fibrotic area goes up with Coxsackie virus B and can be reduced with Astragaloside IV. Finally, in a study from 2019, in a rat model of liver cirrhosis, the rats that received Astragaloside IV had significantly decreased content of aspartate transaminase, alanine transaminase, TNF-alpha as shown here, IL-6 as shown here, and IL-1 beta in the serum of the rats. Also, collagen deposition in liver tissue in the Astragaloside IV treatment group was significantly reduced compared to the model group. The data suggests that Astragaloside IV may not only decrease inflammation, but also fibrosis in rats. Finally, Astragaloside IV has also demonstrated antitumor effects in some preclinical studies. So let's have a look at some of those. The first study investigated the effect of Astragaloside IV on macrophage polarization. Now, why is this important? You all know that macrophages are very important immune cells, and depending on signals from their environment, they can adopt different functional states. The M1 macrophages are pro-inflammatory and anti-tumor. They produce cytokines that help kill pathogens and tumor cells. The M2 macrophages exhibit anti-inflammatory, tissue repair, and immunosuppressive functions. So when the M2 macrophages become so-called TAMs, so tumor-associated macrophages, they promote tumor growth and metastasis, suppress anti-tumor immunity, and drive therapy resistance. So they play an important role in cancer. Now, in this first study, the authors found that Astragaloside IV significantly inhibited IL-13 and IL-4 induced M2 polarization of macrophages. And that Astragaloside IV also suppressed the M2 induced invasion, migration, and angiogenesis of lung cancer cell lines. They also found in an in vivo experiment that Astragaloside IV significantly reduced tumor growth, as seen here, non-treated, and with Astragaloside IV, they also prolonged survival, and they reduced the number of metastases in animal model of lung cancer. The percentage of M2 macrophages was decreased in tumor tissue after Astragaloside IV treatment with 40 mg/kg. In the second study, the authors used a mouse orthotopic lung cancer model to investigate the tumor suppressive effects of Astragaloside IV, and they then analyzed the percentages of various immune cell subsets among the splenic lymphocytes of lung cancer in mice treated with Astragaloside IV, saline, 1-methyltryptophan, and paclitaxel. You can see here that Astragaloside IV prolonged survival in vivo and reduced Tregs while inducing cytotoxic T cells in vitro. So here you can actually see that Astragaloside IV reduces Tregs, while here it actually induces cytotoxic T cells. In the third study, it was shown that Astragaloside IV inhibited migration and cell viability of hepatocellular carcinoma cells via downregulating long non-coding RNA activated by TGF-beta. In the left picture, you actually see that Astragaloside IV dose-dependently decreased long non-coding RNA activated by TGF-beta expression in SMMC771 cells. And here, in this figure, you can see that Astragaloside IV significantly repressed migration of SMMC771 cells. The authors also showed that overexpression here of long non-coding RNA activated by TGF-beta reversed the repressive roles of Astragaloside IV on cell migration. Finally, in a study from 2016, the authors investigated whether Astragaloside IV could enhance cisplatin chemosensitivity. Indeed, co-administration with Astragaloside IV sensitized colorectal cancer cells to cisplatin. So in the previous slides, you have seen that Astragaloside IV has shown promising pharmacological effects in a variety of preclinical studies, but its use as a drug faces a couple of limitations. First of all, poor oral bioavailability. Now, Astragaloside IV is sparingly soluble in water and has very low intestinal permeability, which is of course leading to an absolute oral bioavailability of only about 2 to 7% in rats. This severely limits systemic exposure after oral dosing. Then we have an issue with stability and formulation. Astragaloside IV is tetracyclic triterpenoid saponin, and it is prone to degradation, and therefore it's also difficult to formulate it into stable dosage forms. While Astragaloside IV generally shows low acute toxicity in animal studies, caution is advised during pregnancy due to observed maternal toxicity at high doses. Also, long-term safety data in humans are still lacking. Then, of course, we have limited clinical evidence. Most data come from in vitro and animal models, and I personally could not find robust human trial studies. The current research often uses single disease models, relatively high concentrations or dosages, and that, of course, makes it hard to generalize efficacy and safety across indications. And finally, multi-target complexity. Astragaloside IV acts on multiple signaling pathways, which is, of course, beneficial for pleiotropic effects, but complicates dose optimization and risk assessment for off-target effects. Despite these limitations, Astragaloside IV, of course, has been used in traditional Chinese medicine for many years to strengthen the qi, boosting the immune system or treating other diseases. However, in TCM, it is usually used in form of a tea from the powdered root and oftentimes in combination with other herbs to create some synergistic effects. In terms of the dose, though, Astragaloside IV content in raw root is very low, and the usual dosage of 9 to 30 gram dried root per day may only yield a few milligrams of Astragaloside IV. This is why many people out there prefer these days supplements containing purified Astragaloside IV extract. Most producers recommend 50 mg per day, but again, I could not find robust human PK data, and I would not know what plasma concentrations are actually achieved after a 50 mg per day dose. But I assume that the exposure is far below what's achieved in the experimental studies that I have presented. So while there are many fans of Astragaloside IV supplementation, I would personally like to see more data from studies with humans. For the sake of completion, though, I will put some links to Astragaloside IV supplements into the description, but I will once again emphasize that I do not give any medical advice and that you should always do your own research. Nevertheless, if you have ever taken Astragaloside IV as a supplement, please let me know in the comments what you observed. With this, I'm at the end of the video. I believe there is still a lot of research to be done on this fascinating compound, and I would be particularly interested in some human trials. I will definitely keep monitoring literature and will keep you posted. Besides traditional academic research on Astragaloside IV, I also want to mention Pump.Science, a blockchain-based gamified platform that merges cryptocurrency with real scientific research, particularly longevity experiments. Users can participate in this platform by suggesting compounds tested on organisms like worms, flies, and mice. And Astragaloside IV is actually listed there and is currently undergoing mouse studies. So stay tuned. If you want to head over to Pump.Science to check out some data, of course do that. And please leave a comment. Also, like and share this video as a sign of appreciation. Thank you and see you soon.